Hello students, welcome to EPG Pathshala. I am Dr. Gayatri Ramakrishna from Institute of Liver and Biliary Sciences, and today we are going to discuss the module Basics of Mammalian Cell Culture Techniques from the paper Molecular Therapeutics. This module basically covers the basics of mammalian cell types and how they are routinely cultured in the laboratory conditions. In the area of biotechnology, the mammalian cell culture technology has become an important tool, especially in the area of human health. Health products such as the vaccines, the monoclonal antibodies, the recombinant cell therapeutic proteins all make use of the cell culture techniques. Based on this background, the objectives of the present module are to understand the basics of various cell types and the biology behind it, and the cell culture techniques briefly, together with their applications. Cell is a basic unit of life, be it a prokaryotic cell or a eukaryotic cell like plant or an animal cell. We are restricting this module to mammalian cell culture, which is actually a process of growing cells outside the body. Mammalian cell culture refers to isolation of either cells, tissue, or organoids isolated from specific tissues like the skin, liver, the glands, etc., and further cultivated in an artificial medium. Hence, this mammalian cell culture can be divided as cell culture, explant culture, or an organ culture. During the cultivation of mammalian cells in vitro, that is outside a living organism, some properties are retained as in the original tissue and some functions may be modified or lost with continuous culture. So one has to be careful. Why should there be a need to culture the animal cells? As you all know, cell is the basic unit of life, and understanding the functions and roles of various cell types are crucial for modern biology. Hence, doing mammalian cell culture derived from different organs helps not only in understanding mechanism of how a cell functions, but more importantly, it has tremendous potential in biopharmaceutical areas, which is now called as the red biotechnology. Red biotechnology refers to use of organisms like the mammalian cell cultures for improvement of many medical processes. It includes the designing of mammalian cells for manufacture of pharmaceutical products like the vaccines, therapeutic antibodies, engineering of genetic cures through genomic manipulations, and this includes the cell therapies, and also replacement of animal testing by cytotoxicity tests, especially for cosmetic and cancer drugs. Before we go to the basics of the cell culture, we need to know a little bit of its history. The period from 1880 to early 1990 saw the first developmental techniques to study the behavior of the cells. William Rox and Ross Harrison are considered pioneers for introducing the cell culture techniques. William Rox introduced for the first time techniques of chicken embryo culture. Harrison in 1907 developed the hanging drop method for growing the nerve cells from the frog. The first permanent cell line was developed by Earle in 1943 from the subcutaneous mouse tissue, and in 1951 George Gray derived the first human transformed cell, which is famously called as the Hela cell, and these are derived from a cervical carcinoma. The period from 1950 to 1960 saw major developments in cell techniques. One of the first significant applications of the cell culture arose from the observation that poliovirus could be cultured in simian and human kidney cells. And this led to further refinement of the cell culture techniques and opened more avenues for cellular therapies and biopharmaceutical cell products. The human body contains 30 trillion cells, and the question is, can any cell of the body grow outside the organ? Ideally, the answer is yes. But practically, some cells are difficult to grow, and even if they grow, it is difficult to have a continuous cell culture. So now let's understand what different types of cells are and how they can be cultured. Basically, depending on their culture capacity, one can divide the cells into one of the following categories. Cells which are difficult to divide, for instance, the nerve cells and the muscle cells. Some cells divide when given a cue. For instance, when there is a wound in the skin, the gaping wound is filled by regeneration of the skin cells. Liver is a remarkable internal organ which can regenerate even after it is cut two-thirds. Some cells are immortal and they can continue to divide even in the culture systems. And these include the adult stem cells, the bone marrow stem cells, cells of hematopoietic origin, and the cancer cells, which divide uncontrollably, and hence cancer is a disease condition. Some of the common features of in vitro mammalian cell growth include slow growth of the cells, their complex metabolism, making use of complex media requirements, and the requirements for specialized cell surface for their adherence and growth. Further, some cells are very fragile to grow, and cell culture is most of the time characterized by certain genetic and phenotypic instabilities. Broadly, the different cell types can be divided into the following groups, and this include the primary cells, the immortal or the continuous cells, the transformed cells, and the recombinant cells. Depending on the growth characteristics of the cells are divided into the three categories. Primary cells are those which are taken out from the tissue into culture dish and have a definitive growth span. Usually, these cells follow a typical growth curve of a slow phase, a fast phase, followed by a stop in their growth. The second cell type is the immortal cell lines. The immortal cells grow continuously, but when they reach confluence, they will stop their division. Usually, the immortal cells are non-tumorigenic. However, certain immortal cells are also transformed cell lines which are cancerous in growth. That is, they grow continuously and do not show contact inhibition. The fourth type is the recombinant cells, and these are those cells which produce human recombinant proteins. This is because these cells are then transferred with a foreign gene which usually codes for a growth factor such as an insulin. The recombinant cells are basically used by the pharmaceutical industries for therapeutic recombinant protein production. The recombinant genes of interest, for instance the hemophilia factor X, is cloned and introduced in cells such as the Chinese hamster ovaries or the COS cells. These cells are then selected in antibiotic selections, they are screened, and the best clone is picked and expanded in bioreactors for large-scale production of recombinant proteins. This slide shows some of the most commonly and routinely used cells in laboratory conditions. These include the primary cells such as the endothelial cells, or also called as the human HayFlick cells, the bone marrow cells, the epithelial cells, and the T cells. The immortalized cells category includes the NIH3T3 cells, which are mouse immortalized fibroblasts, HACART, which is a keratinocyte derived from humans, and the immortalized human hepatocytes, which are usually used for many of the studies related with the liver. Similarly, some of the most commonly used cancer cells include the famous Hela cells, the CIHA cells, the C33A. The osteosarcoma class includes the U2OS and the SAOS cells. The hepatocellular carcinoma class includes the HepG2 and the HUH7 cells. THP1 is a monocytic cell line, and the MCF and MDB231 cells belong to the breast cancer family. Cells can also be used for recombinant protein production, and these include the Chinese hamster ovary cells or the CHO cells, the HEC293 cells, 0.6C cell, which is a retinoblast cell, and cos are the monkey kidney cells. It is important to know why it is difficult to grow continuous cell cultures from primary tissues. To understand this question, one needs to understand the phenomenon of HayFlick limit, which is explained in the next two slides. Two scientists, namely HayFlick and Moorhead, in 1961 discovered that the fibroblasts in vitro culture conditions grow only finitely. That is, they have a finite lifespan, and after a few passages, they stop to divide. This stopping of division is a phenomenon known as the HayFlick limit, and this is best explained in the next slide. If we make a typical growth curve from primary cells like the skin fibroblast and plot it in a graph pad, it shows three different phases of growth. The phase 1 is a slow phase, phase 2 is a rapid growth phase, and phase 3 is when they stop to divide. This is indicated here by a red arrow. In phase 3, cells undergo the process of aging, a phenomenon called replicative senescence in vitro culture condition. And replicative senescence is nothing but a permanent growth arrest condition. However, as shown in the graph 2 below, few cells can escape the process of senescence. That is, they can cross the HayFlick limit and can start dividing and become immortal, for example, the cancer cells. HayFlick limit was later explained by the telomere hypothesis. Telomeres are nothing but repetitive DNA sequences at the end of the chromosomes. With each round of division, there is a loss of telomere, and shortening of telomere sets the process of aging or the replicative senescence. When telomeres are too short, it leads to activation of a DNA damage response, thereby leading to cellular crisis, which ultimately culminates in cell death. However, in certain cells, crisis is accompanied by activation of enzyme called the telomerase. Telomerase in turn maintains the telomere length, and the cells keep on dividing, becoming a continuous cell line, that is, they become immortal. And further activation of an oncogene or an inactivation of a tumor suppressor gene can turn the immortal cell into a cancerous cell, which is malignant in nature and can keep on proliferating indefinitely. One of the earliest cancer cells was derived from an American woman, Henrietta Lacks. This cell line is called as the Hela cell. Hela cell is one of the most commonly used cell culture systems in any of the laboratories, as it is easy to grow. This slide describes some commonly used cells in the lab and their feature. For instance, WI38 is derived from the human embryonic lung. It is a fibroblastic in nature and has been extensively used for the production of human vaccines such as the polio vaccine. The second basic objective of this module is to briefly cover the various mammalian cell culture techniques. Under this topic, we would first understand what all are the different commonly used medium for cell culture and what all are included in the basal medium to give a good survival as well as proliferative capacity to the cells. The second part will include the cell culture wares which are commonly available and how they are coated with extracellular matrices for proper adherence of the cell. Various cell culture procedures will be elaborated, and finally in the end we will discuss the various applications of the cell culture techniques for production of viruses, recombinant proteins and monoclonal antibodies. The mammalian cell culture can be done in two ways. Most epithelial and fibroblast cells require a substratum and hence these are cultures of adherent cells. Other cells, like that of the blood cells, the hybridoma cells, and the lymphoblast, can grow even without a substratum, and hence they can be grown as a suspension culture. It is very important to have the right kind of culture medium to grow the cells. Basically, the medium includes an energy source like carbohydrate, for instance the glucose. The media also contains amino acid, of which glutamine is an important component, salts and buffer for osmolarity, antibiotics to prevent growth of bacteria and fungi, and externally this basic cell medium is supplemented with 10% serum of bovine origin, which provides the important growth factors. One can also individually add various growth factors to the basal cell culture medium in absence of serum, and these include the insulin, epidermal growth factor, hydrocortisone, etc. Besides, these days, alternative serum-free media are also available, especially for growth of stem cells. Most of the time, media is red in color. This is because of the presence of a component called the phenol, which is red in color. Phenol is actually a pH indicator, and at pH 7, it is red in color, and as the cell grows, the medium becomes acidic and the phenol color changes from orange to yellow. Thus, phenol is a good indicator of cell growth. Regulating pH is very critical for the optimum culture condition, and this is generally achieved by growing cells in 5 to 10% of carbon dioxide. In a natural buffering system, gaseous carbon dioxide balances between carbonic acid and bicarbonate ions. The cultures with a natural buffering system need to be maintained at an atmospheric pressure at an air atmosphere with 5 to 10% carbon dioxide, which is usually maintained by an external supply of CO2 incubator. The naturally buffering system is low cost and non-toxic. This slide shows the routinely used culture mediums in various laboratories. And also, this slide indicates different cell lines which are routinely used in the labs and the type of the medium in which they are basically grown. Of these, the two most preferred medium in any laboratory are the basal medium DNEM and the RPMI. Besides the medium, another big requirement for the mammalian cell culture is the use of sterile plastic wares. Most of these sterile plastic wares, and especially the dishes, are coated on one side with an extracellular matrix like the collagen, the fibronectin, matrigel, etc. And this is needed for attachment of the cells to the substratum so that they can survive and proliferate. Absence of the substratum leads to a process of cell death called anoikis. Now let's understand the basic protocol for the cell culture procedure. Cell culture or the mammalian culture is usually started from frozen stock of cells. The cells are thawed, the freezing medium is removed, and cells are plated on the appropriate culture dish coated with the extracellular matrix. The cells attach and start to grow. When they are 70% confluent, cells are passed by a process of splitting in which the cells are detached either by trypsinization or by mechanical detachment. The detached cells are then collected in a tube, centrifuged, and plated at suitable density in another culture dish at a ratio of 1:2 or 1:3. Cells can also be stored at this time by controlled rate freezing in a mixture of DMSO and FBS. There are various culture techniques in practice to grow the cultures. This can be in various formats such as a 2D culture, which is basically a monolayer culture with media and the cells attached to the substratum. Cells can also be grown as a spheroid in a 3D culture by use of certain specialized substratum such as the matrigel. Cells can also be grown at an air-liquid interface, and this type of a culture technique actually maintains the stratified tissue organization. There are various applications of an animal cell culture, and these can be categorized into various categories, such as the intracellular activities, which can be used for understanding the process of cell cycle, mitochondrial energetic, process of differentiation, transcription, and protein synthesis. Yet another category is cell-cell interaction. As cells can form colonies, they can be used to study cellular interactions like morphogenesis, motility, extracellular matrix interaction, etc. Cell culture has helped a lot in understanding the biology of signal transduction pathways such as the receptor-mediated, membrane trafficking, endocytosis, etc. Mammalian cell culture can be also used for studying genetic interaction and functionality of the genes by reporter-based assay and by process of transfection. And most importantly, cells are less expensive way to do toxicity testing for new drugs, for process of mutagenesis and carcinogenesis protocols. Stem cell therapy is very popular in disease related to bone marrow defects and immune disorders. And finally, cell culture system is extensively used in biopharmaceutical industries for production of vaccines and recombinant products, which is shown in the next slide. The animal cell culture products is a major component of the biopharmaceutical industries, and this includes production of the vaccines such as vaccine against the polio virus, for hormone production such as the growth hormone for disease related with growth retardation, renal insufficiency, infertility, etc. Interferons produced by the cell culture techniques can be used as an anti-cancer agent and immunomodulators. Plasminogen activators are used for myocardial infarctions, embolisms, and stroke. And blood clotting factors like factors 7, 8, 9, and 10 are used for hemophilia. Animal cell cultures can also be used for growth factor production, and most importantly, the hybridoma technology makes use of the immune cells or the B cells for production of the monoclonal antibodies. Summarizing this module taught to you the differences that are there between the different cell types, namely the primary, immortalized, the cancerous cells. From a primary cell, one can derive an immortal cell by use of the telomerase. And activation of oncogenes or suppression of the tumor suppressor genes makes an immortal cell go towards its cancerous growth. We also understood why primary cells cannot be kept continuously in culture. And this is because of a phenomenon of HayFlick limit. By HayFlick limit, we mean that the cells, after certain passages in the culture, cease to grow. And this stopping of the cell culture is basically due to a process of replicative senescence, which in other words is the process of aging. Our module also discussed about the different cell types and the different cell culture techniques. We discussed the different growth medium, the popularly used mediums such as the DMEM and the RPMI, which are supplemented with 10% fetal calf serum. The medium is also supplemented with antibiotics so that there is no growth of bacteria or the fungi. At times, the media is also supplemented with extra growth factors such as the epidermal growth factors, the insulin, hydrocortisone, etc. to help the cells proliferate much better. We also understood the importance of a buffering system, which in most of the mammalian culture system is the bicarbonate buffer for which an external source of carbon dioxide is used, which is usually 5 to 10%. Briefly, the growth of the cell or the passaging of the cell is done by thawing the frozen stalk, putting the cells into a culture medium, and when they reach 50 to 70% confluency, the cells are trypsinized. The trypsinized cells or the detached cells are then collected into tubes. The pellet is then transferred into another flask following the cell count by using a hemocytometer. At this stage, the cells can also be frozen down for subsequent work. Finally, the module also introduced to you the utility of the mammalian cell culture, especially for functional studies such as the process of differentiation, to understand cell cycle processes, to understand the process of cell-cell interaction, the signal transduction pathways, and also to study genetics or genomics by use of gene transfer techniques. Finally, a vital part of the mammalian cell culture is in the biopharmaceutical industries for production of vaccines. For instance, WI38 is a widely used cell for the polio vaccine. Similarly, the mammalian cell cultures are being used for the production of recombinant proteins such as the growth factors, certain hormones, etc. So with this, I hope you all understood why the cell culture techniques are important and how one can basically start the cell culture techniques in a laboratory condition. Thank you.